Welcome friends, myself Mehtre Suhas, Assistant Professor of ASDS Department from Sadhguru College of Agriculture, Mirsgaon. Today we study the lecture number 5 from 5th semester ASDS course, that is Microbial Quality of Raw Milk and Standards for Different Market Milk. First of all, the introduction about microbial load in milk. The raw milk is never germ free because it has a certain microflora, particularly coming from milk secreting tissues. The other source of microflora, that is, first one is exterior of animal body, that is other area. Then second is utensils used to collect milk. Third is person involved in collection of milk. And fourth one is environment of stables or stalls where animals are housed. The type of microbes found in milk can be divided into four types on the basis of predominant metabolic activity of microbe. First is acid producer, second is a gas producer, third one is proteolytic microbes, fourth one is lipolytic microbes. Then first one is the acid producers. They can enter from dairy equipment, animal feed, polluted water, soil, and certain material of plant origin. These bacteria can ferment certain milk components and they produce organic acid as a predominant and products. The examples of acid producer bacteria are Streptococcus lactis, Streptococcus praemoris, Lactobacillus casei, Lactobacillus bulgaricus, Lactobacillus plantarum, E. coli, and Enterobacter species Micrococcus luteus. These are the examples of acid producer bacteria. The next one is gas producers. They can enter from animal feed, water, and soil. These microbes ferment lactose present in the milk and producing mixture of organic acid firstly. The formic acid present in mixture is further broken down into carbon dioxide and hydrogen. Examples of gas producers bacteria are all coliforms, Clostridium deutericum, and yeast like Torula praemoris. Then third is proteolytic microbes, that is protease. They can enter from utensils, water, and soil in the milk. These microbes produce proteolytic enzymes which are known as protease. These enzymes hydrolyze milk proteins, particularly casein. The examples of proteolytic microbes are Bacillus subtilis, Bacillus cereus, Pseudomonas species, Proteus species, and Streptococcus liquefaciens. Then next is the lipolytic microbes. They can enter from utensils, water, and soil. These bacteria or microbes produce lipase with help of which they are hydrolyze milk fat producing fatty acids. Examples of lipolytic microbes are Pseudomonas fluorescens, Acromobacter lipolyticum, Candida lipolytica, and Penicillium species. Then next is the bacteria. Bacteria are microscopic unicellular which occur principally in the form of spherical, cylindrical, or spiral cells and which produce by transverse fission in milk and its product. The spherical and cylindrical forms are predominant. Most of bacteria vary from 1 to 5 microns in size. Under favorable conditions, the bacteria multiply very rapidly and may double their number in 15 or less minutes. And spore forming bacteria cause trouble in dairy industry because of their resistance to pasteurization and sanitation producers. Next is the molds. The molds are multicellular, differing greatly in most respect from bacteria. Although the individual cells are not visible to the naked eyes at maturity, they may be observed readily by mycelium. They are found in soils, feeds, manures, and poorly washed utensils. The most spores of molds are destroyed by pasteurization. The next is the yeast. The yeast are unicellular but are somewhat larger in size than bacteria. The spores of yeast are readily destroyed during pasteurization. Next is the viruses. In dairy industry, the only those viruses that are parasitic on lactic acid bacteria and known as a starter bacteriophage. The viruses range in size from 0.22 to 0.23 microns. Next is the lactic acid bacteria. In this group of bacteria are able to ferment lactose to lactic acid. They are normally present in the milk and also used as a starter culture in the production of cultured dairy products such as yogurt. Then the lactic acid bacteria, that is lactococci and lactobacilli. In lactococci there are examples is lactococci delbrueckii subspecies, that is lactis, and that is streptococcus lactis. Then second is lactococcus lactis subspecies cremoris, that is streptococcus cremoris. The next is the lactobacilli. Examples of lactobacilli is lactobacillus casei, lactobacillus delbrueckii subspecies lactis, then lactobacillus bulgaricus and leuconostoc. These are the examples of lactic acid bacteria. The next is the coliforms. The coliforms are facultative anaerobic with an optimum growth at 37 degrees Celsius. The coliforms are indicator organisms. They are closely associated with presence of pathogens but not necessarily pathogenic themselves. They also can cause rapid spoilage of milk because they are able to ferment lactose with the production of acid and gas and are able to degrade milk proteins, and they are killed by ST treatment, that is ST pasteurization method. Before their presence after treatment is indicative of contamination. The E. coli is an example belonging to this group. Then next is the significance of microorganisms in milk. First is the information on the microbial content of milk can be used to judge its sanitary quality and the condition of production. Second, if permitted to multiply, bacteria in milk can cause spoilage of the product. Third, milk is a potentially susceptible to contamination with pathogenic microorganisms, and precaution must be taken to minimize this possibility and to destroy pathogens that may gain entrance. Then fourth one is the certain microorganisms produce chemical changes that are desirable in the production of dairy products such as cheese and yogurt. Then next is the bacteriological standards of raw milk. In the table we can see the standards of bacteriological for raw milk. In not exceeding 2 lakh, that milk grade is very good, and it is between 2 lakh to 1 lakh, that milk grade is good. Then the bacteriological standards between 1 lakh to 50,000, that milk grade is fair, and over the 5 lakh, that milk grade is poor. Then next is the standards for milk in India. In buffalo milk, the designation for all the class of milk is raw, pasteurized, boiled, flavored and sterilized. In buffalo milk, the minimum milk fat percentage should be 6%. In then the minimum SNF percentage in buffalo milk should be 9%. In cow milk, minimum fat percentage should be 3.5 to 4% and SNF 8.5%. In goat or sheep milk, the fat percentage should be 3 to 3.5 and SNF should be 9%. Then in standardized milk, the minimum milk fat in standardized milk should be 4.5% and SNF should be 8.5%. Then next is the recombined milk. In a recombined milk, the minimum fat percentage should be 3% and SNF should be 6.5%. Then next is the toned milk. In a toned milk, the fat percentage should be 3% and the SNF should be 8.5%. Then next is the double toned milk. In double toned milk, the milk fat should be 1.5% and SNF should be 9%. The next one is the skim milk. In skim milk, the milk fat should not more than 0.5% and the SNF should be 8.7%. These are the standards for milk in India. Then next is the microbiological standards for raw milk. First one is the direct microscopic count. In the table we can see the count per ml and bacteriological quality of that milk. When there is less than 5 lakh count per ml, that milk's grade is good. Then second is the count per ml is 5 lakhs to 40 lakh, that milk grade is fair. Then third one is the count per ml is 40 lakh to 200 lakh, that milk grade is poor. And then next is the fourth, that milk grade is very poor. The next one is the standard plate count. The count per ml is not extending 2 lakhs, that milk grade is very good and is between 2 lakh to 1 lakh, 2 lakh to 10 lakh, that milk grade is good, and it is between 10 lakh to 50 lakh, milk grade is fair, and over the 5 lakh, that milk grade is poor. Okay, thank you.